Yes. Hi, everyone. I'm Saori. As Kevin kindly introduced, I am the newest assistant professor at the Genome Sciences Department over there. And I just started this April. And I'm having fun interacting with many other departments in addition to the Genome Sciences. So today I would like to talk a little bit about how we can integrate the genetics data with single-cell genomics data to define the causal mechanisms of the common and rare diseases. And if you are interested in this talk, I'm very happy to talk more about my research directions. So just reach out to me, and also it's fun to be interactive. So just interrupt me anytime during the talk if you have any questions or if you need some clarifications. So as Kevin introduced me, I started my career as a physician, specifically a rheumatologist who sees patients with autoimmune diseases. And the most common autoimmune diseases we see is this picture, rheumatoid arthritis, in which patients gradually develops their painful joint symptoms in their hands and foot. And this disease is actually quite common, affecting around 1% of the general populations worldwide. So you may have someone in your family who are affected with this disease, or like who may know someone who are affected this disease. And one of the first questions we always ask whenever we see these kind of autoimmune disease symptoms is if anyone in their family has similar symptoms. And we know this question is quite effective in diagnosing the patients with autoimmune diseases, because we know that rheumatoid arthritis and other autoimmune diseases are highly heritable. So if you have an affected parent, your relative risk of RA becomes 3 times higher than the usual population. If you have affected twin, that becomes 6.5 times more likely to be affected by rheumatoid arthritis. So it's quite clear that it's heritable. You see that even if you have affected spouse, it doesn't quite increase your risk, but it's actually a little bit increase your risk of RA, which may talk about the environmental factors is also playing a major role in the pathogenesis of the rheumatoid arthritis. To me, as a physician, RA is a really an ideal or a good place to treat because there are many drugs that target specific molecules within the inflammatory pathways of the rheumatoid arthritis pathogenesis. For example, you can have B-cell targeted anti-CD20 drugs, or you can have T-cell targeted CD80-86 inhibitors, or fibroblast targeted JAK inhibitors. So if we know which pathway is affected in which patient, then it could be really nice that we can target that therapy for a specific patient. However, when I see these treatment strategies in my clinical training, that was not at all the case in this era of like genomics and data sciences. We usually pick one drug quite arbitrarily. It's really quite arbitrary that like something looks promising, then just pick it and try it, but it may not work. And then this is written obviously in the guidelines that you can try it 3 months, and if it doesn't work, just change it to another thing that looks more promising. But there's no order of the things to be tried in the guidelines, which is like crazy. And until we find the most effective treatment, in this case, IL-6 receptor inhibitor, it's really common that patients has to tolerate the painful joint symptoms more than a year. So it's not so effective in finding the most effective treatment immediately after we diagnose the patient. So I always wonder if we can study more about the genetics and mechanistic insights into the rheumatoid arthritis, then we may guide more effective therapeutics. So that's how I got into the field of the genomics and statistical genetics to somehow I can contribute more to the targeted and mechanistically informed way of treatment in these patients. So today I'd like to talk a little bit about the current status and also challenges in translating disease-associated genetic loci into the causal mechanisms that we want to know about to improve the patient care, and how that can be approached by using the enhancer gene maps, what we call as enhancer gene maps. And we will introduce some method that we developed to define the accurate enhancer gene maps in cell type and tissue specific manner. And also I will introduce the use case of this map to understand the genetic loci that can tell us about the biology of the autoimmunity. And then I finally introduce my ongoing work after I came to the UW about how we can integrate this pipeline of non-coding alleles function in rare diseases. So let's get started with introducing the current challenges in translating the disease-associated loci from the genetics into the causal mechanisms. So I think most of you have at least have heard of the word genome-wide association studies, or GWAS. But just to get everyone on the same page, I just reintroduce these concepts again. So the GWAS is how we can systematically identify the disease-associated genetic loci, which is a quite simple statistical method. In GWAS, we collect many patients' genotype data, and also healthy controls genotype data. It's like the orders of 100,000 of individuals. And then we take just one genetic variant at a time and see if the frequency of this variant allele is different between cases and controls. So in this case, A allele is 80% in the patients, but 40% in the healthy individuals. It could be suggested that this A allele may increase the susceptibility for the RA, and we can formally test this by the logistic regression framework. And so if we do this genome-wide, we get this kind of representative code, famously known as Manhattan plot, because it looks like the Manhattan buildings in the New York City. There are high buildings across the board. So the x-axis is each genetic variant's position on the chromosome from 1 to 22. And the y-axis is the negative log 10 p-value for the association with the genetic variant with the disease. So as each dot, which is the genetic variant, becomes higher in this plot, that indicates that this genetic variant is more highly associated with the disease risk. So this is one of the first projects I published when I started a postdoc for rheumatoid arthritis genetic studies. This is large consortium international efforts, and we identified more than 134 disease-associated loci that has consistent effect on RA patients across the globe. So this is really great that we have a lot of signals across the genome-wide loci that is associated with the disease risk. However, what we find quite challenging to interpret these associated loci is that 90% or more than 90% of the GWAS loci act through non-coding mechanisms. So if the variant is in a coding genome, we can at least estimate that the allele G to A change may lead to the arginine, the protein amino acid sequence, to the lesion by using what we learned in the high school triplet code, so that we can at least estimate that the amino acid sequence can be changed that can lead to dysfunctional protein conformation or something like that. And AlphaFold or alpha missense have done a lot of improvement in this field. So the coding genome, it is more straightforward to interpret the genetic variance function. But as I said, 90% in the non-coding genome, there is no such thing as a triplet code. There's no consensus grammar regarding how these genetic variants affect the gene regulation, which gene is targeted across long distance and in a specific cell type. This grammar regarding how these genetic variants affect that gene expression is really unknown. So we have to figure out that grammar from that data. So the problem we look at from the GWAS, if you look at the single locus among these 130-something genome-wide significant loci, we get this kind of situation. So again, in this plot, this is called a regional plot, where each dot is again a SNP or a genetic variant, and the x-axis indicates the SNP position across on the genome. For example, this is from the chromosome 4 region, and the y-axis is the GWAS significance. And you can clearly see here that many variants, like 20 or so variants, are associated with the rheumatoid arthritis at the same significance. This means that these variants create a haplotype, and we inherit these variants together as from our common ancestor. So if you look at the GWAS statistics alone, maybe this variant is functionally affecting the disease risk, but the others are merely associated with the disease or correlated with the disease because these variants are in a same haplotype or in correlation. So we can't distinguish the causation from the correlation because of this haplotype structure that any genome loci has. So in short, fine mapping or narrowing down the causal variant is not a trivial problem after we do the GWAS. And even after fine mapping the causal variant, it is not straightforward to identify the causal gene, which is affecting the disease risk. So in typical locus, in addition to the fact that we have many candidate variants, we have many genes that is surrounding the significant SNPs like gene A, B, C, D, E, and F. And we can just pick and choose what is most probable as a mediating causal gene to cause the rheumatoid arthritis. There should be some method or prior knowledge about by having this genetic variant, which gene's expression is affected, which is the target causal gene identification problem, which is not also trivial. So to summarize the current status or challenge, even after we do a lot of GWAS to find causal loci, there are a long way from the GWAS loci to find the causal variant, causal gene, and affected cell type and tissue, and affected pathway, ultimately to cause such complex disease of rheumatoid arthritis. So this is the challenge we have in the past 5 or so years after the wealth of knowledge from the genome-wide association studies. But we think that what we call enhancer gene maps can provide a hint to solve this problem. As I said, the GWAS causal variants are enriched in non-coding region, especially in the gene regulatory regions, which mediate or regulate the gene expression. So, and typically the regulatory regions are called the term like enhancers, which enhances the gene expression, or promoters that initiate the gene expression. And if you have a genetic variant within a functionally active enhancer or regulatory region, then the transcription factor, which is a protein necessary to initiate the gene expression, is inhibited to bound to this motif that usually the TF's transcription factor can be bound. So the gene expression of the target molecule is decreased. And this decreased expression may cause the rheumatoid arthritis or other complex diseases. And in this case, you can map the causal variant within functionally active enhancer and also causal gene that is mediating the disease risk. So if, as a wish list, if we have an accurate enhancer gene connection in addition to the GWAS summary statistics, then we can use this map and also use the GWAS statistics to find the causal variant within functionally active enhancer and also define the causal gene that is mediating the disease risk. And understanding the causal gene is a really important problem because if we understand the gene at this locus, this can be a therapeutic target to treat the patient by inhibiting or activating the molecule. So it looks like this is a promising approach, and actually there are many previous efforts to create such enhancer gene maps by using what we call bulk tissue data. For example, one of the common method or gold standard method the field is using is EpiMap, which correlates the epigenomic activity or enhancer activity and gene expression outcome across the donors to define the correlation between epigenome and gene expression relationship to find the links between the two. And the other model is called ABC model, which uses the epigenomic activity, like enhancer activity, and also physical contact between the enhancers to the target gene so that you can link the enhancers to the target gene. So these are one of the most popular methods that in the field is using to annotate or interpret the GWAS loci. But both of these methods are using the bulk tissue data, meaning that you just sample the tissue and grind them and send them to the RNA sequencing data at the donor level. And this is likened as a smoothie because we grind everything as together for the single observation. So first, it may obscure the cell type specific nature of the gene regulation. If the enhancer gene regulation is really limited to a specific cell type that is important for a specific disease, then aggregating everything, grinding everything with the other cell types might dilute that regulatory signal. And also practically, it requires a lot of samples to do a robust association because each one observation at the bulk tissue data is at the donor level. So we have to have hundreds of samples to associate the epigenomic activity or enhancer activity with gene expression to obtain the robust links between the two. So to approach this problem simultaneously, we thought that maybe single-cell data, single-cell multiome data that is recently becoming available is a good data set to work on. So single-cell data, unlike smoothies, is a bowl of fruit salad. So you can look at individual single cells. So obviously, you can more have a granular understanding of the cell type specific gene regulation. And also practically, we don't have to have hundreds of samples to do this similar thing at the single cell association studies. Instead of doing the association at the donor level, we can now associate the epigenomic enhancer activity with gene expression across thousands of single cells, even if we have only a few donors. So in context, we created a method called SENT, or single cell enhancer target gene mapping. So in this method, the data that as used as an input is a single cell multi-ohm data. Multi-ohm, what's the multi means is that we have two measurements. One is ataxic, which indicates where the genome is accessible from the transcription factors that I mentioned as a putative enhancer or gene regulatory element. So the ataxic, we can introduce some sequencing adapters that can specifically sequence the open regions or accessible regions from these transcription factors that we can identify the enhancer elements. And the other modality is the RNA-seq, and which is obviously quantify the gene expression within individual single cells. And what we want to do is to associate the chromatin accessibility or enhancer activity from the ataxic, colored in pink, and with the RNA gene expression on the y-axis so that we can robustly identify the links between regulatory elements or enhancers to their target gene like this. If we have such correlation, then we can link the enhancer activity with the target gene expression. And once we create this accurate enhancer gene map, then we can overlay with the GWAS summary statistics to define the causal variant within functionally active enhancer and also target gene that is mediating the disease risk. So this is the goal of our project, to create accurate enhancer gene maps to fine map causal variant and causal genes. So I want to introduce more about the statistics behind these enhancer gene maps by SENT. The problem that we have to tackle is that ataxic and RNA-seq and the single cells are unique and different from the bulk tissue dataset. So if you ever look at the single cell data in our session or something, you will see this kind of matrix, sparse matrix. So each dot is 0 rows are the genes and the columns are the cells. So you can see that most of the elements in this matrix are zeros. So it's sparse, obviously sparse. And also for the elements that is not zeros, you can see that it's ones and twos and four. So it indicates that these are count values. So this is quite different from the bulk RNA sequencing data, where we have normal distribution of the expression values and chromatin accessibility. So if we want to associate sparse, ataxic, RNA-seq data, then linear correlation would not work because it's not normally distributed. But we don't know yet how these genes are parametrically distributed. But we thought that this is sparse, so we tried Poisson regression, which would handle the sparsely distributed data effectively. So we associated the gene expression count from RNA-seq with peak accessibility from the ataxic, which accounting for some technical covariates such as batch effect by using the Poisson regression model. But when we first tried this, we observed that in now simulated data where we shouldn't have any significant association, we still see significant deviation of the p-values beyond the uniform distribution of the p-values, which indicates that we have uncontrolled type 1 error, false positive associations. And we spent quite a lot of time digging into this phenomena, why Poisson regression doesn't work. And we find that actually some of the single cell gene expression is actually highly expressed and highly variable, even if it's a single cell data. And in that case, the Poisson distribution assumption of the mean variance equivalence and also the sparse distribution wouldn't hold, which resulted in the false positive association if we use the Poisson regression. So the problem that we have to solve is that not all the genes have the same distribution across the board. So the parametric distribution is different in each individual genes. But we don't want to change the model for each gene, which is like too computationally intractable. So instead of going forward with a parametric approach, we added bootstrap procedures, which is non-parametric way to estimate the error in the beta coefficient here we used for the Poisson regression. So in our framework, we first do the analysis using the full data, the gene count, RNA-seq, and peak accessibility to have the single beta estimate. And then we do bootstrapping the cells, meaning that we resample cells again and again with replacement so that slightly different but very similar data set, thousands or tens of thousands of data sets, and we repeatedly do the same association analysis so that we have empirical distribution of the beta, and then we can see the empirical significance of the data beta from the null hypothesis of the beta equals 0, and also we can get the confidence interval so that we can obtain the p-value from the from this distribution, the empirical distribution of the data. And once we took this strategy, we see that our statistics become well calibrated under the null simulation, which now precisely follow the uniform distribution of the p-values, which is estimated under the null associations. So the final model of our SEND method using the cell by chromatin accessibility matrix from the ataxic and cell by gene expression matrix from the RNA-seq, because this is multi-omic experiment, these cells from the ataxic-seq is exactly the same as the RNA-seq from the cell. So there's one-to-one correspondence between the two matrices. And then we associate the gene expression from the RNA and peak accessibility from the ataxic with accounting for some covariates in a specific cell type X. With using the Poisson generalized linear model and also bootstrapping procedures to identify the significant links or correlations or associations between the enhancer or promoter activity with target gene expression so that we can use this to fine map the causal variant in genes from the GPAS. So I hope this was clear to you. Yes.
Yeah. Question is, you mentioned when you the method might help about highly expressed genes. I wonder do you think that could be a over-dispersion issue?
Yeah.
That's the case with negative binomial.
Yeah, we tried a negative binomial model, basically we did the same experiment of this by using negative binomial model, and this inflation became a little bit better, but it's not completely becomes null. So I think even with the negative binomial model, wouldn't fully capture the weird distributed highly expressed genes. Sometimes these elements have like thousand. So I think it's just too outlier to even assume the negative binomial model. So not a single parametric distribution can fully capture the full like gene count distribution, which is like my observation. We could probably be asked to jointly use the negative binomial with bootstrapping, but it is much computationally intensive. So we avoided that because like it's astronomically difficult to computationally track. Yeah.
To do parallax between GPU.
Yeah, it's univariate, one gene with one peak.
How many tests?
It's like millions of tests. So, but we constrained our peaks to be within 500 kb from the gene body. So it wouldn't be genome wide, but it's still a lot of tests.
So what's the way for functional?
Yeah, we just use the Bonferroni correction in FDR, basically correction.
You mentioned the sparsity
Mhm.
of many zeros. Have you tried, for example, zero inflated to account for structural zeros?
Yeah, so zero inflated model wouldn't so different from Poisson regression, and it would account for the sparsity of the data. But in our case, the Poisson was covering that part. But the issue was more highly expressed genes, which was surprising but interesting.
Okay, well, you just robust.
Sorry, what?
Robust stuff.
Oh, yeah. We tried that. We tried like every possible things that is out there. And I think I tried that and also quasi estimate for the confidence interval, something, but none of them worked.
Maybe that also maybe the issue is not as much overinflation. It's not about inflation, maybe it's about the model, the mean model itself. Because I wonder why it doesn't work.
So what do you mean?
Yeah, I wonder if the issue was truly over dispersion or
Over dispersion plus, I think, too highly expressed observation, kind of outlier issue. Two things. Yeah.
All purposes contain also variants. I assume different variants within the peak could disrupt the transcription factor finding in different ways.
Mm-hmm.
Do you see any value in including that information?
So like using the genotype information or something.
Not only links the protein accessory to gene.
Yeah, exactly. And that's what I really am interested in doing, that once such data becomes available. But at the point when we started to like develop this method, the maximum data was from 10 donors or something. So, but now there are like emerging efforts to generate the multiome data in 100 order of samples. Then given the effect size, I think it may become now possible to look at the genotypes effect or interaction effect on the gene regulation. Yeah, so now we were happy with that model. So we applied our method to 9 available single cell data, and 8 of them were publicly accessible data. And the other one, the first one is also now publicly available data to associate the enhancer gene activity in different cell types. The data is mostly for the immune cell types because I'm interested in autoimmune diseases, but also includes the brain, and pituitary data sets that were publicly available at that point. So just a recap. We look at all the ataxic peaks that is colored in yellow and find the significant association with peak accessibility with target gene expression within 500 kb. And if there is some association, we identify them as scent peaks, which is colored in green. And then we looked at the coefficient, how strong these chromatin accessibility or enhancer activity could affect the target gene expression as a function of the transcription start site. And we see that this beta magnitude decays as we get far away from the transcription start site, which is biologically very reasonable because we expect that the promoter regions, which is around 1,000 base pairs, which is very close to the TSS, has the largest effect on the gene expression, whereas the distant enhancers, as we get far away, has more weaker nuanced effect on gene expression. But these weaker enhancers are where the most GWAS variants reside, so it is really important to capture these weak effects. And then we wanted to validate our method by using two experimental data sets. One is the CRISPR flowfish, which is experimentally perturb the regulatory element by using CRISPR technology, CRISPR-I, and then see if truly the gene expression is repressed by using RNA-phage. So like ground truth enhancer gene linkages. And the other one is called H3K27 association high chip, which is identifying the enhancer promoter elements that is physically connected proximity linking method. And in both of the experimental dataset, we see that our enhancer gene linkages compared with the negative associated peak linkages are more enriched in experimentally validated enhancer gene linkages, which we can see that the odds ratio from the Fisher exact test exceeds 1. And then we want to use this scent method to GWAS interpretation. So we first wanted to see truly if our enhancer is functionally active enhancers that more likely to have a GWAS causal variance. And one way to define the GWAS causal variance is from the statistical fine mapping of the GWAS variance. For the easy loci, we can statistically define a single causal variant. And this can be used as a silver standard if to look at the causal variants from the disease GWASs. And we used FinGen UK Biobank data to collect many disease-associated variants. So both in FinGen left and UK Biobank right, we see that scent enhancers in green harvest much more causal variant from the GWAS than just taking the ataxic peaks alone. And moreover, we find that our enrichment for the GWAS causal variants are much higher than previously developed bulk tissue-based method that I introduced, like EpiMap or ABCMap that is currently used in the field. And we thought this result is quite impressive because these bulk tissue enhancer gene map required hundreds of samples to obtain enhancer gene linkages, but our scent method can be applied even from a single donor if we have thousands of single cells. That indicates that our method have a statistical power to reach to the large-scale consortium effort. So even if you are interested in a specific disease that would never be studied in a large consortium, you could, if you have access to the patient tissue data or something, then you can run this method in a single lab. So just to introduce the use case of our approach, so we go back to the GWAS that we ourselves conducted that I highlighted in the Manhattan plot in the first part of my presentation. In this locus on the chromosome 4, again, each dot is a SNP. The y-axis is the chromosomal position of each SNP, and the y-axis is the GWAS significance. So clearly in RA and also other autoimmune disease, type 1 diabetes, there are many SNPs that are associated with the disease with the same strength. So even after statistical fine mapping, it is impossible, just impossible to define the causal variant from the GWAS statistics alone. But now we have the enhancer gene maps from many cell types and tissues by scent. We identified that these accessible chromatin regions that were identified in T cells from rheumatoid arthritis patients were harboring a single causal variant for this locus, which is the most promising candidate for the causal variant. And this enhancer is not as active in other cell types like B cells and myeloid cells. By the way, T cell is the most relevant cell types in the rheumatoid arthritis pathogenesis. And also, what's interesting was that if you look at the peripheral blood samples from healthy individuals' T cells, then this activity is not as strong as the patient's samples T cells. So having the patient sample, having the relevant cell state is critical in defining the functionally active element having the disease variant. And moreover, we were able to identify that in this locus, this RBPJ, which was 200 kBRA distal gene, was the causal gene that is mediating the disease risk. And in my postdoc paper, when we first published this RA GWAS, we picked different gene because the common practice is just pick the closest gene from the lead SNP for the GWAS loci, but it just changes our understanding or biological interpretation of this GWAS by picking just the closest gene, in this case, which was not the causal gene. But the RBPJ gene we identify from scent is a transcription factor critical for immune signaling, and RBPJ's knockdown in mice resulted in abnormal regulatory T cell phenotype, which is the break of the immune activation. So it makes sense that if you have a variant within this Treg pathway, you may have caused the autoimmune diseases. So in this use case, I think I was able to convince you that through our method, we were able to start from the unknown GWAS locus to identify the causal variant, causal gene, RBPJ, and affected cell type and tissue, which was actually T cells from RA patients' joints, and affected pathway of regulatory T cell, and ultimately to cause the complex autoimmune disease of rheumatoid arthritis and type 1 diabetes. So to summarize this part, we created a statistical method, single-cell enhancer target gene mapping or SENT, and it defines the enhancers with functional importance that are enriched in GWAS causal variants, common GWAS causal variants, and it outperformed previous bulk tissue-based method, and also it was useful for understanding the common disease loci from GWAS. So, yes, no worries.
So in the figure where you showed the signal for T cell in rheumatoid arthritis patients stronger than PBMC, I wonder if you just look at PBMC in healthy individuals, would you be able to identify like subpopulations of T cells, for example, memory T cells, assuming they won't, but although they won't be as many as in patients?
Yeah, I think so. Yeah, so I think in inflammatory T cells, there are an expanded population which has more strong association with this variant or enhancer to the target gene, but that population is less represented in the healthy PBMCs. And we can't figure out which cell population then we should be looking at, because if we do this analysis in a very specific cell state by cell state, then we will reduce the statistical power by reducing the numbers of cells we can have. So if we have more larger scale data set, I think that's a really interesting direction that we can pursue. Yeah, so having talked a lot about the common disease to introduce our non-coding enhancer GMAP utility, I am now interested in if we can use this method in the rare disease space, which is much less explored. So non-coding causal areas in rare disease is much less studied. And rare diseases itself is a challenging field because even if that more than 60% of the rare diseases have striking evidence that the genetics are the causes, we don't have yet more definitive molecular diagnosis for more than the 60% of the patients. So as I worked in the clinic, it was very awkward when patient comes in with severe symptoms, and we suspect that, oh, this patient must have some genetic causes, and we take their samples and send it to the whole genome sequencing, and the results come back, and nothing is found. And how can I tell that information? Even if we have a clear suspect that the genetics is a cause, we cannot tell them that this is the cause of variant. And if it's not diagnosed molecularly, then we don't have a clue how to treat them. And one of the reasons why we have like 60% of these patients undiagnosed is that the research has mostly exclusively conducted on the coding regions, because the available data historically for the rare diseases are using the whole exome sequencing data, which is just focusing on the coding regions, protein coding regions, or gene panels looking at the specific genes with non-causal variant. So you cannot have a bandwidth to look at the non-coding variants. But now we have much more availability for whole genome sequencing data, or even long-read sequencing data. So it's now really an exciting time to look at the rare non-coding variants. And indeed, there are such examples. For example, in this published paper by others showed that these recurrent non-coding variants in a specific intronic region of enhancer function has a tissue-specific regulatory effect on the HK1 gene in the pancreas that causes severe congenital hyperinsulinism, basically causing low blood sugar in babies. And we also have such example in our sent paper. So this is an accessible region across T cells, B cells, and myeloid cells at the promoter of the IL-10RA gene, which is an inflammatory gene harboring the non-benign variant registered at the CRIMVA rare disease cohort, causing the autosomal recessive inflammatory bowel disease, so inherited severe form of the inflammatory bowel disease. So I started to be more interested in looking at the rare diseases. So where to start? When we look at the statistics, it seems like the neurological and developmental defects are the two of the most common types of the rare diseases. So I got interested in this area to start, and maybe later autoimmune inflammatory diseases because of my pure interest. And one of the most important moments of the human neurodevelopmental diseases is in the gastrulation period, when a single-layered blastula reorganizes into 3 germ layers, which you may remember in the biology class: ectoderm, mesoderm, and endoderm, and from which all the human adult tissues and organs can arise. So these reorganization is critical for normal and abnormal human development. But despite this importance, these stages of embryo are very difficult to study because the fetal tissue to study this period is virtually impossible to obtain. It's too early to recognize that we are pregnant, so any aborted tissue cannot be available for such early stage of the human development. So it was a black box period, even though it's quite important to understand the normal and abnormal human embryogenesis. But now we have a very exciting experimental system which can faithfully simulate the earliest human embryogenesis up to 28 days post-conception, just capturing the critical gastrulation period, including, by using the induction method, using the retinoic acid and other chemicals from the pluripotent stem cells like iPSCs or ESCs. And this is in collaboration with Nobu Hamazaki staff, who is also a faculty at the Genome Sciences and OBGYN. So now we are collaborating with Hamazaki Lab and started to analyze a time series of single-cell multiome data of differentiating gastroid model to define the functional impact of the non-coding rare disease variants in the very early stage of the human embryogenesis. And as we can see here, we successfully started to identify the most important differentiating cell types in time series manner. And we do see some changing accessibility or changing enhancer activity over the time course of the development. And very excitingly, we observed very interesting example of non-coding variants harbored in such chromatin accessible regions that is causing the severe developmental disease. This is one example. The red bars are the recurrently reported variants in the green bar causing what is called Emberger syndrome, which is a disease causing the lymphedema, leaky lymphatic vessels. So your legs can become larger because of the leaked fluid due to the defect in the lymphatic endothelial cells and causing the dysfunctional junctional integrity. And we see that these variants are strikingly enriched in the accessible enhancer, which is specifically accessible in the endothelium in the early embryogenesis. So this is greatly help us understand the causal mechanisms in a special type specific manner in the most relevant time stage of the embryogenesis. And this is a stem cell based technology. So we can engineer, for example, we can do CRISPR experiment to perturb such enhancer elements that we can nominate from the in silico analysis, and then ask quite interesting questions that no one could have asked, like how these chromatin status can change by the perturbation and how that can disturb normal gene regulatory programs. And beyond the cis gene expression that I comprehensively talked throughout this talk, we can go beyond the single genes expression. We can actually study more complex disease phenotypes like cell lineage decisions or morphology of these embryos by doing these perturbation experiments. And our dream product from this analysis is that we can create such catalog of enhancer element in specific cell type, and we can propose some gene expression perturbation prediction from our experiment, so that if the clinical people pick up some variant, then you can look it up and see what's the possible effect this variant could cause. So with this, I'd like to thank first of all the patients and participants and donors of these precious samples, and especially for the final project, Winston, my postdoc, has been done a lot of amazing work. So I wanted to thank, and Hamazaki Lab, Nob, PI, who developed this awesome new model system, and also my previous advisor, Shomo, for co-developing this method, and happy to take any additional questions.
Maybe I can ask one. How many SNPs, if you were to give a ballpark number, how many SNPs are typically in whatever method you're using to define the peak regions? Am I thinking like 1 or 2 or like 10?
For each peak,
Yeah, like
it's quite narrow. It's only 200 base pairs on average. So you only have one SNP for each peak or 0. So it's simple, yeah, and effective.
What made you start looking at the non-coding regions? Like, what made you start looking at that?
Sorry, what?
What made you start thinking about the non-coding?
Yeah, that's a great question. I think I was struggling throughout my PhD. I was doing the GWAS, and I have to make supplementary tables whenever I have to wrap up the project and manuscript. And one of the most stressful supplementary table is put the statistics from the GWAS, and we have to put some gene names on that supplementary each row for the GWAS locus. And I can pick the closest gene if I am lazy, but I was so unsure about if I'm putting that correct information. And if it's in the coding region, I am so confident of putting that gene name on the supplementary table. So that's how I got more interested in non-coding region, because it looks like many people are struggling with, including myself.
also have a question. So you have this Manhattan plot, and then there are some key genetic hits. And then later, for one of the example, you have probably have the PBMC and then the diseased cells. And then you see some difference in signal. And then later example, you just show like just not stratified by disease status, just I guess it's the overall population signal. So what different information you can get from like this differential signal for the ATAC versus?
I guess you are referring to this plot versus this plot or not?
Yeah. For this one, since you're not stratified.
Yeah. So this is like practical reason. If I see something specific to the disease, I thought it was interesting, so I separated the data set, but we could do this in an aggregated manner.
What information you get from the disease stratified? Do you only look at differential signals and then you apply your scent method?
Yeah, so we apply our scent in each data and each cell type. So the statistics are from each data cell type combinations. And we can compare these significance values across different data sets and different cell types. And it's very interesting that GWAS variants most likely to be in specific enhancers, cell type specific enhancers. And sometimes it's more specific to the disease samples. And I think I mentioned to Guanhao previously, but it's more likely, I think the reasons why it's disease specific is that beyond this T cell, I think there's more fine-grained cell types that is most important for this gene regulation. And having that disease population more expanded in disease tissues have given us more power to detect this association in disease data. And if you look at the healthy data, then you don't have such expanded population. So just looking at the T cells is not enough to find this correlation structure.
So here you have scent separately for RDT side.
Yeah, exactly.
Thank you for the great talk. I usually want to go back to one of your first slides where you showed the relative risk for RA patients with different, like, for example, parent infected and siblings. It seems like there's quite a bit of variation even for like parents supposed to share the same feature coefficient as parent sibling. But that makes me think about what's the role of shared environment in this. But when I was trying to affect the spouse, the relative risk is actually not that high.
Yeah, so I don't have a clear answer. If you want to like more formally assess the heritability, like decomposing the heritable versus non-heritable, like environmental factors, the twin monozygotic and dizygotic twins can have the more accurate estimation because they share the same environment, but the degree of the shared genetics is different. And that study estimates the heritability of RA to be 60% or something. Yeah, so the remaining 40% or so would be environmental something.
Okay.
Arbitrary question. And so the sense is like correlation between ATAC and the gene expression. It's not necessarily like causal. Right? So do you think it's reasonable to apply like linear randomization methods, which will give like more causal effect?
So for the scent, I don't use any genotype information, so it's just a map. And we are sort of more believing that it's causal relationship because the central dogma tells us that epigenome first changes that causes the transcriptomic changes that causes protein function. So like when we do GWAS, we believe that it's causal association because it won't have any reverse causation problem. The genome is what we inherit it doesn't change in theory, and the disease would always follow the genotype. So this time relationship give us more confidence about the causality. And in this sense, analogy is the epigenome would precede the transcriptomic change, which give us a little bit more confidence in causality.
I mean, yeah, the causal ordering is clear, but maybe there could also be confounding.
Yeah, for example, the other way this could cause the just correlated peaks is when two peaks are co-accessible, meaning if this chromatin becomes active, then this should always accompany this activity that happens biologically. Then even if this one is causal and changes the gene expression, because this is also becoming active, then this may also affect the seemingly associate with the gene expression. So that's a possibility.
Yeah.
I think you're right.
To have the genetic as the instrumental variable that's correlated with the ATAC accessibility.
But we don't have the genotype data, or we don't have the power to do that. Yeah, I think rather than like saying Mendelian randomization, I think we can do chromatin accessibility QTL to see if the genotype changes the chromatin accessibility, and if it's corresponding, there is corresponding EQTL expression quantitative loci, then I think the relationship becomes much more clearer.
Yeah, I think it's like a co-validation or Mendelian.
Oh yeah, yeah.
All right, let's thank our speaker one more time. Once again, we don't have seminar for the rest of the quarter, but we do have a statistics symposium. For those of you from students, I was told to, if you still wanted to join, to email Deb. I think there's still for students, rates, or not entirely sure what the details are, but if you're still interested, you didn't sign up, please email Deb, and hope to see you guys there on Zoom.